Hello and welcome to our presentation, Deep Sea Learning, AI for Conservation, a talk about the work being done by students in the Cloud Computing for Big Data CDT in collaboration with the Marine Megafauna Lab, an internal research group over at Newcastle's School of Natural and Environmental Sciences. Our colleagues from this lab are interested in studying and monitoring local populations of cetaceans, so we will cover the what and why of cetacean conservation research before going into the three deep learning PhD projects. So that's deep learning for the collection of data, deep learning for the analysis of image data and automated photo ID, and deep learning for the analysis of audio data and ID using signature whistles. The team from left to right are Cameron, Georgia and myself, Kirsten, from the CDT. The next Tom Hanks castaway-looking guy is Matt Sharp, a PhD student from marine biology. His supervisor and our co-supervisor, Professor Per Begren of Marine Science, Professor Nick Wright of the School of Engineering, and Dr. Stephen McGough from the School of Computing.
I'm Georgia Atkinson. I'm a third-year PhD student within the School of Engineering and also on the CDT. I have a background in maths and statistics, and my work focuses on applying deep learning techniques to underwater bioacoustics.
Hello, my name is Cameron Trotter. I'm a third-year PhD in the School of Engineering. I did my MCom at Newcastle between 2013 and 2017, and I did the taught part of the CDT between 2017 and 2018. My work focuses on automatic photo identification using deep learning, and I'm currently on the hunt for internships.
I'm Kirsten Richardson. I'm in the second year of my PhD. I originally studied psychology and neuroscience before doing a conversion master's in computer science and then joining the CDT. My work focuses on automatic underwater vehicle control using deep reinforcement learning, and I'm currently on the lookout for internships.
Now, we're probably going to say the word cetacean a lot during this presentation, and if you're like me, when I first started this PhD and have absolutely no idea what the word cetacean means, just think dolphins, whales and porpoises. So we focus on cetaceans because the monitoring of marine ecosystems is becoming increasingly important there's a increased awareness of things like climate change and pollutions of the waters. And one way that we can monitor the water is by monitoring the inhabitants of the water. So because cetaceans are top predators of the food chain, this makes them really, really good overall health indicators, because if they are doing well, then there's a good chance that everything below them in the food chain and under the water is also performing well too.
As part of our research, we collect our own data, so we do this within the North Sea off the northeast coastline. So we go out on our boat, which is moored in Blythe, and go all the way up to Cresta, Northumberland, and all the way down to Whitburn and South Tyneside. Within that, we also encapsulate the marine conservation zone, which is highlighted in the lighter colour, and we also have 3 underwater microphones called hydrophones within that area, which collects long-term monitoring data. There are several species of cetaceans which reside within the northeast coastline. For our research, we're interested in dolphin species. We have white-beaked dolphins, which typically live offshore, but within the summer months come inshore to feed with their young. And we also have bottlenose dolphins, which reside within the area all year round. We use 3 different methods for collecting our data. The first is if we go out on the boat and if we see any dolphins that day, we will collect above water images. So we will try and collect pictures of the fins and any body markings that they have. We also collect underwater video. You can see here in this image we have Kirsten bravely hanging over the side of the boat with her hands in the freezing cold North Sea with a GoPro on a stick filming any dolphins as they pass under the water. If we have then collected enough above water images and underwater video, we will turn off the engine and put a hydrophone in the water to collect any dolphin audio that we can. This is used to supplement the long-term monitoring data that we also collect. Since 2019 we've done 46 surveys off the northeast coastline. Sadly, due to COVID last year, we restricted how many surveys we could perform. We've collected over 6,000 above water images, 11.5 hours of below water video, and over 8.5 months of continuous hydrophone audio.
Moving on to how we can apply deep learning to this important kind of research, starting with data collection. 2 methods of data collection that we do not currently use but plan to use in future fieldwork seasons are autonomous underwater vehicles and unmanned aerial vehicles or drones. Drones and AUVs have become increasingly popular in marine mammal research because they are able to capture data that's different to what can be captured from a research vessel. For example, aerial footage captured with a drone provides a bird's eye view of a sighting and is perfect for sizing an animal with methods such as photogrammetry, and it can also be easier to obtain a count estimate from aerial footage than it is from doing it at sea level. The use of AUVs is particularly valuable for behavioural study because it's possible to capture a much broader range of behaviour when you're not limited to filming and recording relatively near to the surface and in the immediate vicinity surrounding the vessel, as is done currently. So at the moment we have a GoPro camera on the end of a pole that we submerge in the water just over the side of the boat, whereas here the AUV can follow the animal and even dive down subsurface. So the usefulness of this method is really evident in the literature, with research groups capturing never before seen behaviours. A quote from the group that used an AUV designated turtle cam is that AUVs offer a revolutionary tool to scientists investigating the subsurface behaviours and habitat use of marine megafauna. And this study and a number of others, such as the earlier shark cam study, first tag the animal with a large transponder tag and then use send-receive electronics such that the AUV is able to locate, follow and film the tagged animal. Our approach to using AUVs is slightly different. It doesn't involve tagging the animal, which is less invasive and avoids all the costs and difficulties involved with tagging. In phase 1, where we are now, we don't technically have an AUV but an ROV, a remote operated vehicle, that has been engineered in-house by Professor Nick Wright, and in this phase the video footage being captured by the onboard camera for later data analysis is also being live streamed to a feedback display held by the researcher, who can steer the vehicle either from a research vessel or perhaps even from shore. We think it's going to have a range of about 1 mile, via the floating radio control box which the system is tethered to. And in phase 2, we cut the cord, if you like, and lose the RC box, and instead of the footage live streaming to a display, it instead streams to a small computing device sitting in some waterproof housing on the actual vehicle, and will serve as input to a neural network saved on the computing device, a pre-trained neural network that will output those same steering commands that the human operator could send in a closed-loop, truly autonomous system. So the problem that we're tackling here with deep learning is one of online motion control, and not only that, but motion control in a way that the system follows a moving target, so active object tracking, and with visual input alone, so from pixel data. And this kind of problem is slightly different to the problems we're used to seeing in computer vision, like classification, where the network receives an image and outputs a category label in this kind of one-off decision. Here we've got this sequential decision-making process where the network is outputting a decision at each time step, and the input to the network at time step 2 is influenced by the decision at time step 1. And because there's this dependency between output and input at the next time step, we can't start with a fixed data set of training images. Rather, the data is collected from actually running the network and having it make decisions and interacting with what we call the environment. And this problem setup is well suited to a paradigm of machine learning called reinforcement learning. And whilst there isn't time in this presentation to cover how reinforcement learning works and how it's different to supervised deep learning, there is a demonstration here that it at least does work, and the QR code here takes you to a blog about this early project we did called Cube Track, which was about training a network to control the movement of the blue cube such that it follows the purple target cube. And this of course is not in real life but in a simulator, and the idea is to step up the complexity of this simulation until we're as close as possible to the real world setting of an AUV in water following a dolphin. And then at that point try to bridge the sim to real gap and get the physical AUV tested in a water tank with a model target.
So now let's take a look at some other uses of deep learning in this space, specifically my work in automatic photo identification. So what is photo identification? So photo identification is a method used by marine biologists. So essentially what happens is during fieldwork out on boats, if they encounter any cetaceans, they collect photographic data of that encounter. And then these photographs are then taken back to the lab, and the individuals are identified based on prominent markings, usually on the fins. And this kind of study is really useful for things like population dynamics and ecology health assessments. But there are some issues with manual photo identification. Kind of the top two. Number one, it's very time consuming. So an average fieldwork season, it will take about 3 months to go from raw photos to an ID catalogue that's usable. And it's also prone to error. There's a high risk of mislabeling because the identification process requires the researcher that is performing it to have seen the individual previously. What kind of things are the researchers using to identify the individuals? Number one, they're looking for things like nicks, so kind of any notches or parts of the fin that have came away, either naturally from other predators or cetaceans or through anthropogenic conflict. They're also looking for scarring, so this usually occurs when the animal has interacted with things such as boats or propellers or anchors. Any long-term scratching on the fin, as well as any pigmentation. This usually occurs from things such as disease. So when building an automatic photo identification pipeline, it's important to think about how we can split this problem down. So we can split it into 3 parts. Number one, a detection. So given an input image, we need to detect if there's a fin in it. 2, segmentation. So if there is a fin, can we remove that from the image for processing? And 3, classification. So given a segmented fin, can we determine which individual the fin belongs to? And to understand how difficult of a problem this is, we need to break this down further and understand the concept of coarse, fine, and extreme fine grain. So in a coarse grain task, such as the detection task, we're only detecting on a very high level label. So in the image on screen, all 3 objects would be given the high level label of dog. In our task, we would be labeling things dolphin. In a fine grain task, we go down a bit deeper into the hierarchy, so all of the objects would be given poodle rather than just straight dog. If our classification task was fine grain, our labels would be things like either bottlenose or white-beaked. We'd be looking at a species level rather than an individual level. But we are looking at individuals, and so this is what we call an extreme fine grain problem. Because we're looking for very, very minute details to differentiate between our classes. So in the image on screen, we would we would be identifying based on the dog's names, and that's very similar, or exactly the same as what we're doing here, just with cetaceans. Having neural networks perform extreme fine-grained classification is still an open area of research. Most current research focuses on things like interspecies differences and dog breeds. But if you are doing extreme fine-grained research, chances are you're doing things like facial recognition or person re-identification. And these conditions are usually quite sterile, so it's why passport photos have such stringent rules. In contrast, we're working in the North Sea, where conditions are anything but sterile. So now that we understand the problem, let's take a look at the solution. So this is the automatic photo ID pipeline that we've been developing. So again, we kind of go through the stages. So we take an input image, we run it through a detector to see if there are any fins in there. Then we take those fins and run them through a segmenter. We clean them up. And then we run it through a photo ID system in order to determine the individual extreme fine grain identification. Looking at each component in more detail, the detector is just a standard Mascar CNN with a ResNet 50 backbone that has been pre-trained on Microsoft's Common Objects in Context dataset and then transfer learned onto our own dataset. The Mascar CNN is used because it provides pixel-wise mappings for objects, which is very, very important for the segmentation algorithm. Because we need to remove as much background as possible to be able to aid the classification system. Once we have some detected fins, they are then passed to the segmenter, where we remove the fin from the image on a pixel-wise basis. So this reduces the computational expense of further operations and also helps remove a very, very noisy background, i.e. the sea. So by removing this noisy background, we help improve the chances of classification because then the classification algorithm can't get confused with waves and things like that. And there's no deep learning here. This is all just performed using OpenCV in Python. Now for the hard part, actually identifying the animals. So in order to implement an automatic photo ID system, we need something that's capable of, one, accurately locating the identifiable information in the segmentation, where that information might be very, very minute. We need to, two, compare that found information to potentially very few class examples, whilst being invariant to things such as rotation, occlusion, lack of features on the fin. So not only is this an extreme fine-grained problem, it's also a few-shot learning problem. So luckily we can use a thing called Siamese neural networks to help us get around both of these problems. I don't have time to go into the details of Siamese neural networks in this talk, but in essence we take a high dimensional input such as an image and embed this down into a low dimension, essentially giving us a set of numbers which represent the input. This embedding can then be plotted into some multi-dimensional space, and the network is trained to cluster these embeddings in the space such that images of the same class have close embeddings and those of different classes have highly different embeddings. This allows us to work with classes where we have very few examples, as in theory we only need to have one example image in order to form a cluster. We can also work with classes unseen during training, as the network should be robust enough to plot new classes into their own space, and when we're working with unseen classes such as free-roaming animals, this is highly important. Note here that we don't predict a classification per se, but we rather predict how similar an image is to those previously seen. There's so much more to talk about here. If you're interested in Siamese neural networks, I've wrote about them quite a bit. You can scan the QR code to read that. And if you want to know more about specific parts of my system, please come and speak to me during the poster session.
Moving on from automatic photo identification, we will now look at acoustic identification. Like Cameron, I'm also looking at identifying dolphins, but I'm going to do this audibly instead of visually. Dolphins produce a variety of vocalizations, including things like whistles and echolocation clicks. They use echolocation clicks for foraging, and we're not entirely sure what all of the whistle types mean. Some species of dolphins, like bottlenose dolphins that we have in the North Sea, produce signature whistles. You can think of these as unique vocalizations to help identify individuals. Think of this like a dolphin's name. They say their name audibly, for example, Georgia, 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 so dolphins in the pod around them know that they're there. A common technique when looking at audio is to convert audio signals into spectrograms. These are a visual representation of audio. If we first look at the left-hand plot, we can see we plot time against amplitude. You can see we have an audio signal there, but we don't entirely know if this audio clip contains any whistles or not. If we just have a listen to this clip, the high-pitched vocalizations within that clip are the dolphin whistles. So you can see you clearly can't see this in this plot on the left-hand side. But if we now look at the spectrogram on the right-hand side, you see we now have time plotted against frequency, and then the colorization is the amplitude. So the darker the color, the louder the sound. If we just have a listen to that audio clip again, you can see now from the high-pitched vocalizations here, they're clearly the dolphin whistles within the spectrogram, so the dark lines in the spectrogram. So now I'm going to look at how we actually detect the signature whistles. So split this into 3 stages. The first stage is to detect any type of whistle that we see. So we go through and look at the spectrograms and find the start and end points of each of the whistles. We then move on to stage 2. This is where we cluster the whistles to determine if the whistles are similar or not. And the third stage is to determine if the whistles that we've clustered are signature whistles or not. So we do this by using a signature identification method that is commonly used in marine biology, that looks at the timing information of the whistles. So before I go on and talk about how I detect whistles, I'm going to talk to you about a couple of issues I have with my data. I have over 8 months of continuous audio which is not labelled, which is from the long-term monitoring data collection method that I mentioned before. I have a very small amount of labelled data from the data that we collect when we're on the boat, and even from the data that I have, a lot of the data is really noisy. So this is due to things like anthropogenic noise, noise of boats, and it's also very noisy depending on the weather. Due to these issues with the data that I've mentioned, when we're looking at the whistle detection phase, the first thing that I do is create synthetic data. I take noise that is from the audio recordings and try to create fake whistles on top of that noise based on shapes that I've seen from papers. Then train a detection model on this synthetic data. So currently we feed in the raw audio, convert it to spectrograms, and then train a convolutional recurrent neural network. We then run the detection model on some of the unlabeled data from the long-term monitoring, and get the predicted labels outputted. At this stage, we get a lot of false positives due to the synthetic data not being a good representation of the data that we had collected. So we go through all the predicted labels and pick out the correctly labelled audio and put this back into our whistle detection model to retrain. We also feed the correctly labelled data back into our synthetic data. So we're looking at using a generative adversarial network to enhance our synthetic data so it looks more realistic to the data that we have collected. Coming soon. Very soon I'm going to start working on being able to cluster the whistles and then move on to trying to automate the signature identification method that I mentioned before to detect the signature whistles. The whistle detection model is also going to be applied to streaming data. In the engineering department, C Lab have developed an acoustic network gateway buoy which live streams audio from the sea back to the shore. Thanks for listening. If anyone's interested further in what me, Cameron and Kirsten are doing in our work, come catch us at the poster session.